And as you might have noticed, I'm pretty casual about most things, so interrupt me at any point. I've stuck a QR code and then just a bit.ly URL for like the published to web version of this slide deck. So if you want them in your hands so you can flip back and forth, I found that to be super useful. My first two slides, I'm going to skip right over. It's my abstract, and then I stuck a couple citations in. And so I also have like a list of the papers that are shaping a lot of my thinking that I'm talking about, just because I get frustrated when folks give talks and I can't remember what the references were. So that's yet another reason to get into the slide deck if you're interested in the details of some of this. Cassandra's introduction was really nice, and it's a stretch to say that I understand anything about the impact of scenes on D1, but that is what I've been obsessed with lately. But I first wanted to start with this is my timeline, a bit of introduction, since I don't know many of you personally, and this just shapes my perspective as all things do. I was born in 1973 in Albuquerque, New Mexico. That becomes relevant in a second, the year. That's also the year that the Nobel Prizes were awarded for some of the foundational discoveries of MRI. That Lauterbur and Mansfield shared the 1973 Nobel Prize. And when you go home, if someone wants to know like which part of the city you grew up in, you always ask which high school did you go to. And so I went to Sandia High School, graduated from that in 1991. Here's me as a high school student with my feet up on the desk with my dad's computer. Mice had not been invented yet. And so when I learned to love things related to computers, it was to get attention from my father. Math and computers was how I got attention from my parents. And we played early versions of like Zork and Find the Lumpus and Tic-Tac-Toe and Star Trek using just like arrow keys on a keyboard. I did my bachelor's in physics at Michigan State because I was born thinking I was going to be a physicist. I worked at 3M for a couple of years after my bachelor's. I had moved to Minnesota to marry the wrong man. That's a whole nother story we'll tell some other time. So I needed to get a job here with just a bachelor's degree in physics. And I worked in fiber optics. And first I worked in developing a non-traditional large core plastic fiber optic as a neon alternative light source. So I was just wholly a physicist into optical characterization of things. And then I was supervising summer students who were getting paid more than I was because I didn't have any letters after my name bigger than a bachelor's. So I decided I needed to go back to graduate school. And I joined the home team at the time and did my PhD here in the neuroscience program. Because I figured if I was going to go back to school for another 5 years, I was going to learn something completely different. And yet, neuroscience of all of the life sciences was the one that to my eye looked the most like physics, and it had optics and eyes see light. And so you can see my career path was like not well planned, but it worked out great. I got here and I found out that we are the Rome of magnets, and I just really, really love putting people inside MRI scanners and seeing pictures of brains. The point of my talk today is lately I've gotten very frustrated by what functional MRI does not tell us about brains, but I'll talk more about that later. I was lucky enough to get the job that I have now when I was ABD, when I interviewed still as a PhD student, and they were kind enough to defer it for a year so I could go to New York University and get some. You've got to see more than one institution in your life. And so I really enjoyed a crash course at NYU working with David Heeger. And then I came back to take this job where I've been in the golden handcuffs ever since. And so this is some headshot I had to do for some teaching award. Really, I wanted them to let me put the image of the pineapple on the monitor, but they thought nobody would understand what they were looking at if I put the pineapple. So that's my brain and my pineapple, which I love scanning. So you can see from this trajectory, physics and the magnet part of my life is kind of my first love because I love things that are predictable and go in straight lines. And before I get too deep into it, I just want to do shout out to my lab. These are folks. Well, every time you take a picture, it gets out of date in 2 months. Victor's moved on to do a postdoc at Northwestern, and Jesse's out at Maastricht doing a postdoc in non-optic sight. Look up Jessie Breedlove if you want to just get to know an awesome person. She studies vision in folks who can't see, and she's a very empathetic researcher who's very interested in how the brain creates images. And Joe Emerson, a lot of his work is what I'm talking about today, but I'm blessed with a really fun to work with lab. And as I sat down, I realized I'd left out a couple grants that funded some of the stuff I'm talking about, but a shout out to my funders, and then just acknowledgement that the University of Minnesota doesn't always have a clear history for how we took resources from our neighbors. So there's the context I'm working in, and I get weirdly entranced just looking at a tree, because the more I learn about vision, the more I understand that we don't really see what we see. Like, you look at a tree, you feel like you're seeing all of those leaves, but you're really not seeing them in like the literal photons hit my eyeballs. And then like, we just don't have the level of resolution to process that many leaves all at one time. But you have the sense of the complexity of the tree without literally seeing all the details. So as we know, since Hubel and Wiesel in the 50s, visual information is first processed in the back of the brain, primary visual cortex, neurons that love isolated features and can sense color. We have area V4 is kind of what I'm highlighting here as a intermediate level of the brain that does more complex features than just edges, and is very selective for color. And as we travel forward along the ventral visual stream, you find neural circuits that are selective for specific objects that you've learned to recognize. But also, as you go up across the dorsal part of the brain, you find regions that modulate strongly on where you're allocating your attention and what you're planning on doing with those things. And if we get all the way up to the front of the brain, we can start thinking about the goals you hold. So this is maybe 20 years ago, we talked about these things the way I just described it. There's these different parts of the brain that process these different aspects of your visual experience. And really, my point today is we can't isolate all of these processes, even doing what I'm about to do, which is like zoom into V1 and use tasks that really try hard to isolate the specific tasks that we know that primary visual cortex is very, very good at doing. My premise is that the neurons in V1 are like immediately driven, biased, contaminated, informed by all the rest of this stuff that's going on in your brain. And so I'm having a mid-career crisis these last couple of years, as I have to kind of ditch my original plan of starting at the back of the brain and working forward as we understood the building blocks of vision. And I'm wrestling with the fact that primary visual cortex is always also representing your goals, your expectations, your prior experiences. So I'll explain that later. My premise for this talk, the reason I keep showing these pictures of these moths, is that the stimuli that I'm using in the laboratory that I'm talking to you about, which are very boring, are analogous and useful for understanding these much more interesting stimuli. Here's a dark moth and a light moth on the bark of what to me looks like a birch tree. And because of where the dark moth is sitting, it has feature contrast, so it sticks out. It's easier to see than the light moth, who has more wisely positioned itself to avoid predation and is better camouflaged. So I'm talking about low-level features like color, lines, orientation, and their role in making things easy or hard to see. And so when I show stimuli like this, I'm talking about analogous to the black moth is an orthogonal sinusoidal grading, where I have some kind of a circular target that is easily detected against its grading background because of an orientation change, whereas there's a more subtly camouflaged target when things are parallel. So we've studied this paradigm for a long time. And what I'm talking about for the next couple minutes is all the ways in which the responses surprise us when we think we know what's happening. But I'll start by talking about we think we know what's happening. So neurons in primary visual cortex, this orange thing is a neuron, imaginary. And as it's got a receptive field, so according to my color key in the lower left part of my slide, it likes things that are almost vertical. And as I increase the contrast, the difference between the black and white parts of this circular patch, as I increase the contrast, the neuron increases its firing rate. This is an electrophysiology paper from Tony Movsham's lab. In the low contrast regime, it has this accelerating nonlinearity. And then in the high contrast regime, those responses start saturating because at some point you can see it. There's no point wasting extra action potentials encoding that. Toward the end of the talk, I'm going to talk about the computational modeling framework that we're using to try to simulate all of these neural mechanisms I'm talking about. And this transition from supra-linear low contrast regime to a sublinear high contrast regime is an important aspect that we're trying to capture. Stimuli never appear in isolation, so there's going to be some kind of a surrounding context. Here is the orthogonal surrounding context that makes these things easy to see. Some neighboring neuron with a different preferred orientation is going to be interacting with our target neuron that's seeing the center at its preferred orientation. It might be a weakly inhibitory interaction. It might be a weakly facilitative interaction. I'm showing this little tiny white neuron as some kind of a intermediate interneuron that's helping them interact. But if that surrounding context is matched along some low-level feature dimension, such as orientation, that lateral inhibitory interaction is really strong. And that's where we get this surround suppression that I'm going to talk about a lot, that uniform textures, there's no point spending a lot of action potentials encoding everything that's going on in a uniform texture. So it's reasonable that the brain would have mechanisms that would kind of automatically suppress similarity. So that's a rationale for orientation-tuned surround suppression, which, again, I'm using to try to mimic more naturalistic and more interesting things like moths on trees. So how do we measure what's going on in the head? I mean, I study humans, right? I don't have electrodes in heads. I haven't seen a neuron in person since I was a graduate student. So the outputs that I can measure are not the firing rates of neurons. I can measure your behavioral sensitivity. So we do psychophysical measurements on people. And in this suppressed regime, your sensitivity to change is also suppressed. So I can use your contrast discrimination or detection thresholds as a metric for whether or not this output is suppressed. And there's decent literature showing that those two things correlate pretty well, although I'm going to break that in a second. And then the other thing I can do is functional MRI. We've got these really big magnets. I know how to put people in them, and it's really fun to see brains. And I relied heavily on this paper from Barbara Zenger-Landholt out of David Heeker's lab right before I joined as a postdoc. Honking huge surround little targets and measuring the fMRI response to these targets and comparing it to the behavioral response to this annular target. And they did a really beautiful job of showing that the suppression measured by fMRI matched the suppression measured by psychophysics. So the y-axis is serving two functions here. The dashed lines are indicating the behavioral responses, so the sensitivity to change in contrast in that surrounded annulus. And with the surround, the sensitivity to change drops. And at the same time, they did the functional MRI experiment, and this was a very careful design that I admire a lot. And if you do it right, you can watch the fMRI response to the annulus be weaker in the presence of the surround. So I went into this series of studies armed with this confidence that it was important to study surround modulation of V1 responses, because that was like, so we have Gabor elements, they match the receptive fields, we know the contrast response function. Now we're studying how it's modulated by its surround, and once we get a handle on that, we can start putting them together in more interesting patterns and add behavioral tasks. That was my logic. So I launched into this set of experiments studying contrast surround suppression, thinking, this is straightforward, I'll just do the building blocks for vision. But it's more complicated than we would, of course, like it to be, because, of course, we're using functional MRI, and each voxel, even when I push to, like, the highest resolution data I'm going to show you today are 0.6 millimeter isotropic resolution. That's crazy high resolution. But even those tiny-ish voxels have tens of thousands of neurons in them, and they contain all of these populations. So far, I've told a story that's focused on, like, the one neuron that's seeing its preferred stimulus, but it exists in this whole population. Each of them are seeing stimuli that are not their preferred. And then the actual modulation by surround gets super complicated super fast. So the behavioral effect is pretty straightforward. I measure your suppression and perceived contrast as a function of the relative orientation between my center target and my surround annulus. And presuming that when you decide which button you're going to push when you're doing my task, you're relying on action potentials that come from the most sensitive neuron in your brain, which is the neuron tuned to the orientation of the target. The behavior is a pretty nice readout that matches our simplistic thinking really well, where as I rotate the surround away from the center, I see your behavioral response show less and less of a suppressive effect with relative orientation. But as I try to think about how that's going to predict my fMRI activity on a fine scale, I need to remember that my fMRI voxel likely includes, it does include many, many different neurons with different preferred orientations and connectivity patterns. And it often includes the surround as well as the center. So I need to think of these interactions like the surround is suppressing the center, but the center is also suppressing or facilitating the surround. So that's one of the complications of considering the entire population. And then it gets, I'm just adding more neurons into the mix to talk more about spatial scale, because another effect that we've studied that I'm not talking about today, but that we've thought about a fair amount, is the tilt illusion. Again, I've taken a target grading and I've surrounded it by a surround with differing levels of similarity. And if there's a small mismatch in alignment, you get this repulsive effect where the orientation of the central target looks like it's rotated away from vertical one direction. But if there's a big center surround orientation difference, then I get this other attractive effect where the surround pulls the orientation. So this starts telling a really interesting and complicated story. Work out of Tomas Serre's lab has done a really nice V1 intrinsic model that explains all of this by talking about very short range facilitative interactions between neurons with similar preferred orientations, and then these longer range interactions that sum and then inhibit the population by some kind of a weighted sum of the orientation in the surround. And if you put these short range facilitative mechanisms and these long range suppressive mechanisms together, you can predict the tilt illusion. I also love Odelia Schwartz's work, which doesn't rely on V1 intrinsic model and talks about natural scene statistics and probabilities and what's the likelihood that the center and surround belong together. And if you use like a prior expectation for belonging together as a thing that gates the strength of your surround suppression, you can also arrive at an adequate model of the tilt illusion that predicts these kind of more complicated behaviors. And we don't yet have the data or the ability to adjudicate between these two models. And there's, of course, a distinct possibility that both are true. So now I've waded way down into the weeds of thinking about the thousands of neurons in primary visual cortex that are in my fMRI voxel and contributing to your behavior, because I wanted to like make it look messy real quick. So we could agree that it's not simple to study even the most boring V1 behavior like surround suppression. And I'm going to add one more thing to that, also following along the similar logic of Odelia Schwartz's work. This was a paper by Ruben Cohen-Kaigi with Adam Cohn and Odelia Schwartz, looking at electrophysiology data and looking at this higher level gating of these suppressive interactions. And they call it flexible normalization. And the word normalization comes from this white arrow I drew with summation, when you sum some kind of pool of responses, and then you pool that into an inhibitory neuronal population that that gates by division the response. So suppression by division is a normalization is a short word for that. And it's flexible because they had good data from these electrophysiology experiments that whether or not you got the standard suppression by parallel surround, which is shown by these blue circles, we got a blue central target, we've got some kind of a parallel surround of zebra stripes. This is a kind of extreme example. Over in the red, they show like the part of the surround falls off the back of the zebra. And there are more subtle examples where there's like a different texture cue, like between the zebra's cheek and his neck, where your 3D scene segmentation process that is going on somewhere in your brain, which is way outside primary visual cortex. Through your scene understanding, you've decided the center and the surround, they don't belong to each other anymore. So that's referred here as independent or heterogeneous surround. And what they saw in the electrophysiology data, that even though the statistics of the center and the surround might match, when there was a higher level factor that would predict that the center and surround don't belong to each other, they would see some kind of a gating that would actually turn off the surround suppression. So that's like taking this lowest curve in this contrast response function with and without surround suppression from the Kavanaugh paper. That's like turning parallel surrounds into orthogonal surrounds by turning off the suppression, not because the surround has changed to being orthogonal, but because something has decided that the center and the surround don't belong together. So it's no longer advantageous to suppress everything because some kind of an interesting object contrast has happened, so we need to turn off that normalization. So I'm really interested in that and how it happens. And so I've added to this whole messy V1 model this feedback thing called big picture signals. And at the end of the talk, I'm going to nod to a model that proposes that this actually happens by feedback that targets a particular subpopulation of interneurons, which oppresses the oppressors. So we're aware of specific physiological mechanisms that are feedback that targets inhibitory interneurons that themselves target the inhibitory interneurons that then releases the suppression of the population. So that's where I was going with all of these studies, is I wanted to be able to study these mechanisms by which like feed forward signals would automatically cause suppression, and then feedback signals would release that from suppression. But I got stuck for a while on this idea of multiplexing because from this mess, predicting the average response in an fMRI voxel is really challenging because everything's happening all at once and not everywhere. It's in my same voxel. So the neurons are receiving that input drive. They're getting this facilitation and inhibition from local context, and they're also being regulated by big picture context. Hence the basis of my mid-career crisis, because if all of that is going on in a single fMRI voxel, how are we going to interpret our data? Obviously, by doing many, many experiments and using a lot of different stimuli and building computational models that bridge animal data with human data. But it's not a simple endeavor. My moths are appearing again in this slide, just to remind us that the whole point is not to understand sinusoidal gratings. I am convinced that sinusoidal gratings do not, in many ways, reflect the neural mechanisms that serve natural seeing vision. They are a laboratory convenience, so I'm continuing to use them for a little while longer. Okay, to the data. So this was one study that we did in a large, this is one of the grants that was missing off my acknowledgement slide. It's a collaboration with Scott Sponheim, who's a trained in clinical psychology here at the University of Minnesota. He sees patients clinically, he does research, and he's very good at running these large family studies, is what we call them, because he'll recruit individuals who have a psychiatric diagnosis that includes an element of psychosis. So that would be patients with schizophrenia or patients with bipolar disorder, first-degree relatives, and then that enables the study to try to separate genetic liability versus expression of psychotic disorders, and then a population of controls. So we had 20, 30 people in each of these 5 groups in for a study that did both behavior and functional MRI on these stimuli, which you now recognize very well. In the behavioral version of the task, they were never taking their eyes off the square in the middle and using their peripheral vision to compare the contrasts of these central targets. And we were adjusting the contrast of the reference on a staircase to try to match the perceived contrast. So that's where my data come from measuring what is the relative perceived contrast of this central target when it's surrounded by parallel gratings versus things that are rotated a little bit or a lot or are completely orthogonal. And then the behavioral data came as expected, as I've been showing you, that when things were parallel, all of the groups, PBD is patients with bipolar disorder, PSC is patients with a diagnosis of schizophrenia, HC is healthy controls, and then we have relatives of the 2 groups that I'm showing for the sake of thoroughness, but not talking about today. And so in all groups, we saw the expected behavior of more suppression for parallel and less suppression for orthogonal surrounds. What we didn't expect to see was that the patients with schizophrenia, which in about a dozen other studies have always showed weaker suppression. So that's one of the first things you learn about visual perceptual abnormalities in patients with schizophrenia, is that suppression is weaker. Because we had not dropped the contrast of the central target, because a parallel arm of this experiment was also measuring that tilt illusion that I told you about. Since we had a high contrast center, we discovered that our patients performed the same as our controls in the parallel condition. And as the surround rotated away from parallel, we actually saw this reduction in the amount of suppression, so that the reduced suppression only happened when the segmentation cues were very strong. We have a couple of theories for why more work needs to be done on the topic. And one more reason that more work needs to be done on the topic is this thing that really surprised us about acuity. So as we do intake on these patients, we stand everybody up in front of an eye chart using the correction they're going to be using for the experiment at the same working distance of the experiment. And we measure their acuity. We're using a Logmar scale here. So 0 is equivalent to 20/20. And as you move rightward on this first axis, you're getting toward worse and worse acuity. And all of the individuals from all the groups are plotted here in a giant scatter plot, different colors for different groups. The y-axis is M, and I should have explained M. We fit these curves showing the change in perceived contrast with relative orientation as an exponential function, because that's a good fit to these curves. And the amplitude of that exponential function is M. So M is a proxy for the sensitivity to the relative orientation, or like the steepness of the curves. Not steepness, amplitude. It turns out the steepness of the curves didn't differ between groups. If I separated all of my populations into the folks that had great acuity, like, well, not great, but better, like better than 20/30, or worse than 20/30. And you could ask, why did I have so many people whose acuity was worse than 20/30? And I'm not going to be able to give you a good answer to that. On that day, when they showed up in the clinic and they did the eye chart for us, they didn't have the right glasses. They weren't wearing the right glasses for the viewing distance. It was not our design to put people into this task where they would be doing it with poor acuity, but we did. And if we looked at folks who were doing the task with good acuity, separate from folks who were doing the task with lower acuity, we were very surprised. The performance changed dramatically, and the performance changed in different ways for individuals with a schizophrenia diagnosis and healthy controls. For the controls, the acuity changed in a way that you might expect. If I'm seeing these stimuli, which I'm going to go back to the last slide, and if you squint at them, your ability to tell the center and the surround and find that little fiducial mark that I've put between, and your ability to see the gap, if you squint at that, all your segmentation goes away. So if all your segmentation goes away, you expect the suppression to get stronger. And so this black line where the control participants with poor acuity just showed poof across the board, stronger suppression of perceived contrast everywhere. That made sense. What didn't make sense was for the patients with schizophrenia. In the parallel context, their perceived contrast dropped, as you would expect from just blurring things. But as the segmentation cues got strong, and the orientation as well as the gap between the stimuli helped separate the two things, the suppression actually got weaker. So there it is. We have theories. We could talk about them later. We have not pursued this. There's a lot of work to do to pursue this. There's a lot of different reasons that acuity could have this weird interaction between patient groups. But that was just one of my surprises, that to study surround suppression, especially in patient populations, I was going to need to know more about the underlying neural mechanisms, which is why I've backed off from those studies for a bit. And I'm spending more time on computational models these days so I can have more specific hypotheses about neuronal subtypes. So I can come back to this question later with exactly which part of inhibition is changing. The other thing we did in that study is, of course, we put in the scanner, because there's nothing we love better than putting things in scanners. And in this case, they were doing a fixation task, and they were distracted from the stimuli so we could present twice as many stimuli so we could get twice as much data. The behavior, I'm just recapitulating the behavioral figure here on the lower left. And on the right, I'm showing the prediction for the fMRI response, which is that in the parallel condition, we would expect a reduced fMRI response when we isolate. We're pretty good at isolating just the voxels that are responding to the target. And then as the relative orientation increases, we should get a release from suppression, so the fMRI response should increase, and it should increase more for the individuals with schizophrenia, since their behavior also shows a greater release from suppression. So the fMRI prediction was 20 degrees lower than 70 degrees, and just out last week is the fMRI result, which is the exact opposite of the prediction. Not strongly, but convincingly not the answer we expected, that we got in all groups, and most notably in the individuals with schizophrenia, a stronger response in the condition where we expected suppression to be larger. We also have bigger responses in the right hemisphere across the board than in the left hemisphere. I also have no explanation for that, nor am I going to opine on it at the moment. So there's just a lot of mysteries hanging out here, giving me something to do for the second half of my career. But we really wrestled with this, and the thing that helped us most was a paper from Scott Murray's lab, 2015, I think it is, with Anastasia Flavaris, and they manipulated attention. And I'm sorry, these are very tiny, but in each pair of bars, the one on the right is a collinear or parallel context, and the one on the left has flankers that are orthogonal to a center target. And they did functional MRI, and I believe they also did behavioral and electrophysiology, EEG, I mean. And but fMRI is the most relevant to us, when they had their participants attend at the central targets. So attention was enhancing allocating spatial attention selectively to the centers is going to like shrink the summation field and enhance the responses to the targets. With attention on the centers, they got what we expected as a result. Lower response with parallel as opposed to orthogonal context. But if they changed nothing about the stimuli, they changed nothing about position, they changed nothing about where the eyes were, and they just instructed the participants to instead attend to the upper flanker. So you're putting your attention out on what I would have called the surround. It flipped their responses around. This paper is totally worth reading. They have a nice model that's kind of following the Reynolds and Heeger 2009 kind of normalization model of attention. It's kind of like oppressing the oppressors, but it's kind of like enhancing the oppressors. You can model this effectively by looking at where attention is allocated, how attention is increasing either the center response or the target response, how that spreads out to include, and then also how features, depending on the orientation. The key thing here is if you're attending the surround, then you're attending either to a similar feature or to a different feature. And so having attention on the surround also manipulates feature similarity between the attended and unattended regions, and it all makes sense. So I believe what we accidentally did in this fMRI project was by putting attention just on fixation to encourage our participants to hold their eyes still so we could localize those targets. Whatever attention they weren't spending on their fixation task was going to naturally be drawn to what was going on out there in the periphery. And in our orthogonal condition, they would have been therefore allocating some accidental attention to a surround that didn't have a feature that matched the center, giving us a lower response in that condition. So I think it makes sense, but it made me really shy about studying surround suppression in the scanner because of how strongly spatial attention is going to manipulate responses. That didn't stop me from doing my favorite study that I'm going to tell you about as the third and last experiment I'm going to describe. So I'm just going back to my overly complicated illustration of all the different neurons at all the different spatial locations, representing centers and near surrounds and far surrounds and all of the different interactions between them. All of that's happening in a single region of interest. So we went and tried to do this at super high resolution so we could take advantage of depth-dependent functional MRI in order to try to, we're not going to spatially separate all these different neurons in a single, all the different preferred orientations, and we're not going to separate excitation from inhibition. But we can look down through the cortical depth and try to separate out the input responses from the lateral connections from the feedback responses. So that's this premise for a lot of us who are chasing this depth-dependent signals in MRI. Everything I've illustrated to date is conceivably the superficial neuronal population, which would be the output of primary visual cortex up in layers 2 and 3, where you have excitatory populations. I'm illustrating them by these white circles, and inhibitory populations. There's many more arrows you should put on this graph. But you have these interacting excitatory and inhibitory neuronal populations in superficial, middle, and deep layers. And input comes into the bottom part of layer 4 in primary visual cortex, ripples very quickly through all the depths in primary visual cortex. Reporting is out from V1 to V2 and downstream visual areas comes from superficial layers. Immediately, and in the case of auditory stimuli, before that even happens, I just learned the other day that if you have an auditory cue, auditory-driven responses in primary visual cortex can show up even before the feed-forward visually just driven responses. So something over there is going to grab my attention. It's going to potentiate V1 responses before that hurts me, before the feed-forward pathway even gets there. I feel like I should have studied audition. It feels like the winner. But those feedback signals tend to target the deep and the superficial layers and kind of leave the middle layers alone. So you can look for biases towards superficial versus middle responses as an indication of where the signal might be coming from in your surround suppression signal. So that's what we went after with this high resolution experiment. I'm going to show you the stimuli. We had a blank screen. We didn't start out with a blank screen. I will show you why we ended up with a true rest condition. This was originally our intended baseline. We're going to present our target, not surprisingly, where I've been talking about these little peripheral annular or circular locations. That's our surround only condition. We're going to have a just full field grading. I'm sorry you have to look at this for so long, but please do. This would be the maximally suppressed location. This little artifact that's showing up on this slide was not present in the stimulus. There was no boundary that would give you a sense between what was the target and what was the surround. But then we copied this out of Self 2016, out of Peter Rolfsema's lab. If you just do a 90-degree phase shift between the surround, you still have parallel, but you have a very strong boundary cue. So by comparing this phase shifted, we call it ISO 90 condition, against that non-phase shifted version, you can still have a parallel surround, but a nice figure ground segmentation cue. And then, of course, rotation is going to give us a nice, hopefully, release from suppression. And it's nice to have a control between those two that has essentially equivalent edges defining those figures. So that was just a block design. We're doing super high resolution data collection. The participant is engaged in orientation discrimination task. This white circle wasn't actually a circle. There was a grading in there that matched the orientation of the surround, and they were doing 2 interval forced choice orientation discrimination tasks throughout the scanning session. So they weren't paying attention to what was going on in my little peripheral targets. They were just staring at the center of the screen. And even during this baseline condition, there was still a central target. So they were just doing the same foveal orientation discrimination tasks throughout the whole scanning session. And I just wanted to show off some really fun data to play with this. We did gradient echo EPI, 2-second TR, 27 slices, 0.6 millimeters thick, 27 slices. We just had a little tiny slab going through the back of the head. It was enough to catch the V1 region of interest, plus some V2, V3. I'll talk in a second about denoising the data. And then one of the really cool things we did, if anyone in the audience is a high resolution fMRI buff, then you will appreciate the glories of doing a T1 weighted EPI, which is this lighter, crisper image right here. It's still acquired with the same echoplanar imaging distorted data acquisition strategy that the functional data are acquired with, but it's got an inversion preparation pulse in front of it, so I can measure the T1 recovery rate in each voxel, and it gives me that nice, crisp, almost like anatomical quality level. So I can do all of my analysis in the distorted functional space and be really sure where I'm working. We do acquire reference anatomies. We do sometimes see the stria of Gennari, which is what I'm highlighting with those crosshairs right there. And then we assign every voxel in our data a relative depth. And for people who care about the details, we use an equivolume depth solution to try to be most accurate, because layer 4, the input layer, rides up and down in the cortical depth as it goes around the curvature of the brain. Okay, I could talk about methods forever, but I'll stop there. I just want to show off, like, we thought 0.8 millimeter isotropic, which is kind of the standard layer fMRI resolution. We thought it was great until we started seeing 0.6 millimeter data. Now at the 10.5T, I'm not part of the crew that's doing that, but they're getting 0.4 millimeter data. It's really fun to see the brains at this level of resolution. You can't do it with good signal to noise ratio, so you need some wicked denoising techniques. So here's a single volume of what my data look like coming off the scanner. And then if I apply Nordic, which is a denoising technique that was developed here in Minnesota, it just looks for the independent uncorrelated noise in your data. And I swear it doesn't blur the data. It doesn't eat my data. Sometimes it does. You can tell when it goes wrong. But it almost never touches my data, and I just come out of that with a denoise data set that has almost quadrupled sometimes my signal to noise ratio. So that's what enabled doing this experiment at this resolution. I'm going to give very brief results, and I'm happy to talk about more of them later, but I also want to quit talking pretty soon so we can get into some Q&A kind of stuff. The circular targets were localized with independent localizers. You see this oval yellow patch in the back of the brain in V1. It's an oval just because of how cortical magnification is going to stretch things out. So I did PCA on the flattened coordinates of the individual voxels in order to draw a circle at 2 sigma, where 2 sigma is defined by a mixture of the eigenvalues of the SVD that I used to do the PCA to discover this ellipse. I'm showing 10 representative hemispheres from the early half of the data set. Each voxel is plotted as a circle and color-coded according to whether it preferred the center or the surround in the localizer. So you can see some ranges, and we see a few people where we're like, oh, is that really what you're doing with that? But by and large, we were pretty happy with the localization, and these patches ended up being 5 to 8 millimeters in diameter, so pretty big area to work with. So the first contrast of interest, so now my y-axis is the depth through the gray matter. So with each voxel was assigned a location across the cortex space as well as down through the cortex depth, so we could slice our data however we wanted to. And now we're just grouping everything that was selectively representing the target, and we're going down through the cortical depth, and we're contrasting the just the little edge that's introduced by the shifting the phase of the inner and outer gratings. So that would be the figure ground modulation center signal without any rotation going on. And we see a modulation that is not everything we hoped for. We hoped there would be no modulation in the middle of the cortical depth and then strong modulation top and bottom, which would be the pattern we expect from these feedback signals. It's pretty close. And so we're pretty happy. Like here we've isolated a feedback signal that is the figure ground modulation. I'll tell you in a second one caveat for that. But I want to contrast that against the other contrast of interest, which is just the rotation. So the edge is there, and all we've done is rotate the context in the center. And you'll notice these are tiny, tiny signals because I'm subtracting 2 large responses to just hone in on just the signal change that's caused by that rotation. And we see it in restricted to superficial layers, not present at all in deep layers, and weak enough that I'm unconcerned in the middle layers. So this would be the depth-dependent signature of the orientation-tuned inhibition, mediated putatively by these horizontal connections in superficial layers. And we can detect that separately from the figure ground modulation feedback, which has a more all the way through the cortical depth kind of pattern. So we feel successful there, and we feel really surprised here. So I'm going to end on just another giant question mark in our lives. And this is our what we wanted to be the baseline compared to what we expected to be just the most suppressed condition of everything. And first, I'm showing you early results from the first 5 individuals. And at the time, I was analyzing the hemispheres separately. Since then, reviewers have been very unhappy with that approach. And so everything else will be listing individuals, not hemispheres, as my independent sample. The reviewers are not wrong on that one. But it's just so hard to get this data. You're so proud, and every ROI is made independently. But they are not independent because individuals have strongly correlated signals between hemispheres. But the point of this plot is that the surround-only condition where there was no feed-forward stimulus into the receptive field of the neurons gave us even bigger responses than the sine wave grading, which would have feed-forward suppressed, but nonetheless feed-forward drive. We're like, really? So that was the first 5 individuals. We went back and got more. So these are 12 different individuals. Showing all the individual data plus the average response to the surround-only condition, the average response to the uniform grading condition, and then the difference between the two, which in this one way of analyzing it, where we've restricted ourselves to a region of interest in the center that responds better to the center than the surround, we'll see that in superficial layers, that region responds. So it's negative. The iso minus surround is giving us a negative response here. So that means that the very, very, very center region here is responding better to the surround when there's nothing, no stimulus in the present than it is to the center plus the surround. So it felt like a really backwards kind of result. You're looking at this thinking, well, isn't that just blurring of the data? Like fMRI veins blur signal all the time. And I can't say you're wrong. I'm going to show you one more thing, which is a radial profile. So now instead of going down through depth, I'm going out across the surface. And this is in the superficial layers. There's a step in the data analysis I didn't tell you about that we call deveining, where we've removed from the analysis any voxels that are clearly contaminated by large veins. And this is, again, the difference of the uniform grading minus the hole in the grading. And so the location of the edge in this analysis, so the 2 sigma line is drawn in red here, right at the edge of the voxels that prefer the center. The actual edge of the stimulus is between the 2 and 3 sigma lines. So you see this slight bump in the negative response right at the location of the edge. And so the fMRI data prefers the surround everywhere, except once you get down into the center. And so it's here in the a very restricted region of interest, less than 1 sigma. So it's half the size of the red oval that I've drawn over on the side. Inside that region, where there's no reason to believe that the population receptive fields overlap the edge at all, we see this match between the feed-forward suppressed drive and the feedback-only drive. If this were merely blurring from the surround signal coming into the center, I would expect it to keep dropping off and get even lower as we get toward the center. Whoops. So these are my future directions, right? I can't prove, but I believe that the signal that I'm seeing here in the very, very center of my region of interest in response to no stimulus is a feedback receptive field. And I'll give you some evidence for that before we open up for conversation. So this is another paper out of Rolf Summa's lab. This is Lisa Kirchberger's paper from a couple of years ago. They did the same experiment in mice with depth electrodes. And the RF blank condition is equivalent to my surround only condition, and the uniform condition is equivalent to the ISO condition I've been showing you. Here they're measuring multi-unit activity, lots of mice, lots of penetrations, lots of controls on the population receptive field, doing their dead level best to prove that, and there are other figures in the paper, that the units that they're measuring here did not respond to stimuli in the surround when they were presented like in isolation. But if you put this whole huge predictive surround around my target region of interest, you get this delayed but quite robust response, which is something we're all starting to call the feedback receptive field. They did further analyses and to show that this particular response was tuned to the orientation of the surround. Again, a skeptic would want to argue, well, that's just some kind of a blurred response from the surround, but in electrophysiology, the blurring isn't like what we worry about in fMRI. And so that blurring or spreading of signal is most likely mediated by feedback signals that are predictive of the surround, but isn't actually an interesting neural mechanism. Oh, and I skipped the current source density depth profiles here, but for the 2 conditions that had feed forward drive, you see this big bump of current sink in layer 4, and then in the RF blank drive here, but so many other things, like the other V1 responses. So this is starting to be like the signature of feedback drive to primary visual cortex. For the interest of time, I'm going to skip. I have a set of slides at the end that are about computational modeling and the way we're building up that model. I don't want to run long. I can talk about that later. So suffice it to say, I'll skip to the end of it. Here's the end of it. That we're working with a collaborator, Gordon Smith, who's got 2 photon imaging in ferrets. We can look at selective tuning of PV, SOM, and GAD, are 3 different inhibitory subtypes of neurons, as well as excitatory pyramidal cells. Joe Emerson, I can't skip his picture, a graduate student in the lab, is the one actually doing the modeling, supported by Audrey Sederberg. And so we're using this stabilized superlinear network motif to build a sheet that'll let us simulate the kinds of responses that we're measuring. So here's the SSN sheet to simulate. And eventually we want to work this up so we can start putting that like virtual neuron feedback. Here's the effect of attention. Here's the feedback of prior experience. Here's the feedback of expectation and scene surround. So we can start getting kind of a quantitative computational handle on that. But there is not time to talk about that today. So I just want to end on this slide, which is like why I'm obsessed with feedback receptive fields. And another half of my life has spent a lot of time hanging out with people who talk about Bayesian inference and predictive coding. And the image I grabbed on the right, I grabbed out of a StairSearch paper in 2018 talking about how people were closing the loop here, how people use predictive coding to talk about psychosis. This is one account. This is by far from a settled question. But the idea here would be that you have some kind of a prior expectation about the world that's going to come at you through feedback. It's got some kind of like a mean and distribution. Let's ignore the equations. You've got some kind of a sensory input, a sensory likelihood, and then the combination of your prior and your likelihood is just multiply these two Gaussians together and get a posterior distribution for what you're going to perceive, which is in this case, even mixture. You could weight those things, right, of your expectation and your likelihood. The reason I'm obsessed with feedback receptive fields these days is I feel like the feedback receptive field is a great way of naming the prior. And so this physiological evidence for feedback receptive fields could very well be the thing that we want to use in like a Bayesian model of predictive coding for saying, yeah, this neuron, it's got these synaptic connections to the rest of the brain that we're calling a feedback receptive field. That is how you multiply the prior against the sensory likelihood in order to come up with some kind of a posterior, which is what you see. So I will end there, and I would be delighted to take any questions, comments, or fixes to the logic.